السلام عليكم ورحمة الله وبركاته. أنا Alhomam Dabaliz. I'm a second year medical student, and today, inshallah, I'll have a lecture about the introduction to molecular medicine. So, Bismillah, Allahumma la tahmil wa sahla, and let's begin. So first off, I wanted to make sure you understand one concept real quick. Molecular medicine is a science by itself. It is not biochemistry. Biochemistry is a subspecialty in that science. Molecular medicine is the big umbrella, and biochemistry is just a part of it. So the university, when it named this course molecular medicine, it didn't do it all willy-nilly. There was a specific reason for it. And this reason will be explored a bit more in this lecture. This lecture won't be very academic heavy, it won't be a lot of information. So anyone who's trying to take notes, you're welcomed, but there's not much to take notes about. Okay? Here's my contact information if anybody needs it, and I'm in the Premed group as one of the academic committee. So, to start off, I always like to start off with the definition. Molecular medicine has a definition by itself. Molecular medicine's definition is a very well-made definition that will include all what you will ever study in molecular medicine as a whole. Okay? So this definition states that molecular medicine is a broad field that uses different types of techniques, like we have biological, chemical, bioinformatics, whatever techniques. They are used to understand structures of molecules and mechanisms that molecules work in. Okay? After describing these structures and mechanisms, we can identify fundamental molecular and genetic errors of diseases. And that is exactly what us as medical students are meant to know. We are studying this course as a whole as medical students to understand the errors of diseases and to learn how to develop molecular interventions to correct them. Okay? Now, I will take the first half of the lecture on this definition. I'll talk about this definition in detail. I'll be talking about this definition in detail just to make you understand more what is molecular medicine. So starting off, we will talk about these three words: chemical, biological, and medical. Medical is pretty obvious. We are medical students. We will study everything in molecular medicine in a medical context, the diseases and how to treat them. What about chemical and biological? Well, this is what our University of Alfaisal focused on in this course. Molecular medicine, as I said, is a broad field. It has many subspecialties, but the two subspecialties Alfaisal University focused on are biochemistry and cell biology. These two subspecialties are very interesting and very important for us as medical students, much more than all the other techniques that was mentioned in the definition. In Alfaisal, molecular medicine is mostly biased towards biochemistry actually. The reason for that is very simple. Cell biology is a very broad term. It's actually a very broad term that studies the structure and function of the cell. When I say structure and function of the cell, I will think mostly of yesterday actually. Mohammed Jamour, my friend, gave you a full lecture about the cell. And in that lecture, he mentioned a lot about the structure of the cell. But that was in physiology. And that's exactly why cell biology is kind of shafted in this course. Cell biology is studied more in medicine in the context of physiology, because in cell biology we study the function of the cell. So each type of cell has its own function, and that's why we study it mostly in physiology. However, there are some lectures, some functions of the cell that aren't specified to a certain system, like the renal system, the GIT system, etc. And those functions are the ones we study in molecular medicine. On the other hand, biochemistry, the major part of the course you will take now, which studies the processes within the living organisms. These processes are mostly done to ensure the life of the organism. It is not dictated only to humans, of course. This course is the same for humans as it is for every animal, most plants, most even singular cell organisms use most what we will discuss in this course. So, actually, speaking of how we learn more of biochemistry, if we consider that in the first year we will study around 52 lectures of molecular medicine, around 33 of them will be biochemistry, while the rest will be cell biology. That just shows you how much we actually study more biochemistry, and that's why people, when they mention MOL, they mention biochemistry more than cell biology. Most of the people don't even know what cell biology really is. Now, I wanted to talk about something that I really love to learn in science, the origins. How did this science come to be, actually? When we talk about the origin of any science, we need to go back way back in time. And for biochemistry, we actually need to go back to the 17th century. The 17th century was an interesting time in which many of the sciences actually developed. And this science, biochemistry in particular, developed from a simple question. What is it that separates living systems from other systems? Many people wonder this. Why is the table not alive and my hand is considered alive? People at that time had very small knowledge on biology, especially the microscopic level, because microscopes were not invented. And that leads me to the first point, the first time this science was actually, well, kick-started. And that was with this man, Robert Hooke, when he discovered, or invented actually, the microscope. And through it discovered cells on cork, filline. And discovered cells in it. Actually, he named them cells because they look like the cells in prisons. And from that, the whole science kick-started. People started studying the cell. What is this cell? What does it do? And they were not really satisfied with saying, oh, something had a cell, then it's alive. That didn't really satisfy them enough. What is it that made the cell itself alive? Because literally they learned that there are unicellular organisms. So what made them alive? And that's how this science was more kick-started, more and more. The cell was studied heavily, or I want to talk a bit about Robert Hooke actually. Robert Hooke was a very interesting man. He was both a physicist and a biologist, inventing the microscope with his physics knowledge actually. And he was the student of Boyle. Does anyone remember Boyle's law? Yeah, he was a student of Boyle and actually worked with Boyle on that law himself. And he lived in a very interesting time. It's a time where people didn't know what oxygen is. A time where bacteria were demons, and being sick means being cursed. Anything people didn't understand was basically magic. And that's why a man like him, who studied nature as not magic but science, was not very common in that time period actually. So, let's go fast forward a bit. 19th centuries, all right? In the 19th centuries, the knowledge about the cell was accelerating very quickly. Organelles were being discovered, their functions were being discovered, okay? But not only the cell was being discovered here. We don't live in a vacuum. Every science has other sciences running parallel to it. And in this case, another science was actually learning about a new building block of the universe. While the cell was considered the building block of living creatures, atoms were being discovered as the building block of the universe. And that led scientists to ask this very simple question. If cells are the origins of life, the root of life, and atoms are at the root of everything, does that mean that atoms are at the root of life? And does that mean that us, as humans, are basically a bunch of atoms? That was very controversial, actually. Not many people knew how to answer that. And it stayed that for quite a long time, for a century almost. Until a man who was actually a physicist Or before that, let me mention this. In the 20th century, early 20th century, a lot of scientists, biologists, said this quote: "The cell is too complicated that we will never learn how it functions." And, well, they were obviously proven wrong. We know a lot about the cell right now. So much, in fact, that we can make courses about it. We can teach people all day and night about the cell. And we still have a lot to learn, that is correct. But you should never say never in science. Because in that same century, a physicist by the name of Schrödinger, who was very popular for his quantum mechanics discoveries, this man wrote a book called What is Life? And in that book, he stated, without a shadow of doubt, even without really much background on biology and without much evidence to back it up, he stated that without a shadow of doubt, functions have roots in molecules. Every cell's function has a root in molecules. And he stated that especially one function that will definitely have a root in molecule is inheritance. Mendel had already done all his research by that time. Everyone knew that inheritance was a system, and Mendel studied it a lot. But they had no idea that molecules were at the base of this system. So, back to our little definition. We talked about chemical, biological, and medical. What do they mean exactly? Now let's go to the next part. We have molecular structures and mechanisms. This is a big part of what you will study in this course, actually. And the best example I can give of studying molecular structures and mechanisms came right after Schrödinger said his word that, yes, functions are at the basis of everything. Two men, very well known. I think everyone saw this picture before in their textbooks of biology. Francis Crick and James Watson. Those two worked together very hard on discovering the DNA. They studied DNA a lot, they studied inheritance a lot, they studied everything about it a lot. And, well, they found out, before finding the chemical structure of DNA, other people before them found out that DNA are the source of inheritance. It was an experiment that also our biology textbooks really wrote about is it proteins or is it DNA that actually transmit genetic information? I'll not mention it here because I'm pretty sure that we all took it in biology. And we all took this in biology as well. Because these two men used a lot of research from other people, like, for example, Franklin over here, Rosalind Franklin. Now, there's a lot of controversy about this topic. I'm not going to go into that, okay? But suffice to say, they coined in that the structure of DNA is this beautiful little mess. This took researchers years and centuries to find how DNA is structured and how this molecule is structured. That is basically molecular medicine. Those people weren't biochemists or molecular medicine practitioners, no. They were scientists, they were biologists, chemists, even philosophers actually. And still, they had one of the biggest interventions, biggest achievements in molecular medicine. Okay? Now, while James Watson's biggest achievement after this is staying alive until now, Francis Crick has another big achievement that is actually coined to him, and he didn't have any hand in stealing much. And that was the central dogma of molecular medicine. The central dogma is a good example of molecular mechanisms. We said molecular structures with the DNA, but now molecular mechanism would be this. The central dogma talks about how DNA is used to make proteins. And this DNA transcribed to RNA and then proteins. This mechanism also took so much for people to study, and until now it is studied heavily, and you guys in the molecular medicine course, you will take around, I believe, 7 lectures just at this slide alone, just to understand everything that goes on here. You'll also take around 2 lectures on this, and 2 lectures on just the RNA on its own. So yes, everything you see here, you will actually take again in molecular medicine as much more in detail. Now, back to our definition. We finished the whole molecular structures and molecular mechanisms. What about this? The point that actually us as medical students should be most interested in. Errors, fundamental molecular and genetic errors of disease. Now, there's one disease that I believe all of us studied in high school. It's very apparent when you see someone with it, you know it is, oh, he has it. And, well, it has a root. Its root is heavily in molecular medicine. Talking about albinism. Al-Buhaq is a disease where someone just loses his color. All his color is gone from hair, eyes, and skin because his body cannot make melanin. Okay? And melanin comes from an amino acid, that's tyrosine. And again, to study how albinism happens, we need to know the structure of tyrosine before it becomes melanin, and the structure of melanin, all the mechanisms that turn tyrosine into melanin, and all the mechanisms that went wrong when turning tyrosine into melanin, to understand albinism. Now, I could have, of course, put in a picture of albino childs or adults, but I think most of us saw these before, and so I decided to put animals with albinism. They look actually kind of cute, honestly. I love this peacock. But yeah, even animals can have albinisms because 90% of the color in the animal kingdom comes from melanin. So if we have a problem in tyrosine metabolism to melanin, then you do not have color. Of course, the red in the eye is because the eye is transparent, no melanin, it's transparent to blood. This is blood you're seeing. Okay? Lastly, in our definition, we have developing molecular intervention to correct them. Now, another disease, very, very popular in this area of the world and honestly everywhere else. Diabetes, we all know it. Okay? Sukr. Diabetes is 100% biochemistry. It's just biochemistry. Okay? There's a part of it from genetics, but the how the disease shows on a person is from biochemistry, of course. And the number one intervention we use for diabetes as a whole is this, insulin. We just give them insulin shots. Now, how did we reach this point that, oh, he has a disease, we give him insulin? It's not exactly just a question of, do we give him this or this? We have to go through all of this all over again. When we found this one treatment, we had to study insulin as itself, as its functions, as its biological intervention, its chemical origins. You need to study its structures, you need to study its mechanisms, and you need to study why diabetes, well, why does diabetes happen? Well, we found out that, oh, it's because there's no insulin. We studied what insulin does, and then we studied its structure so we can make it in labs. And that is exactly why I really, really like this definition. It takes everything you will study in this course and just applies it in a simple paragraph. Okay? So, I took a while on this. I will not take a full break now. I'll actually keep talking, but I will talk about some random things, honestly. While I was researching for this one lecture, I actually took a while researching for this lecture. I have a lecture around 4 weeks from now, glycolysis. I took way less time researching for that lecture over this, even though glycolysis is 2 hours. But when I was researching this, I watched around 7 hours of YouTube, which was also put on 1.5 speed, so that's a lot of YouTube. Also, read around 5 Wikipedia articles, or 6 of them, I think. And yeah, I read a lot here and there, and one fact that I saw very cute actually is, you all know protozoa, طلائعيات. All know it? Okay. Protozoas were first discovered after the microscope was invented, after Robert Hooke actually died. Okay. So the first protozoa that was discovered is Hydra. It is a animal-like protozoa. Yes? So when it was discovered, a term was used for the kingdom of protozoas, which was animalcules. I really like that name, animalcule. But later on, they discovered plant fuels, basically. So they realized that, oh, the name animal fuel isn't very accurate, and that's why they changed it into protozoa, as it is kind of well, not as advanced as us plants or animals or such. But I really like that name, honestly. I would love it if it stays. It's animal fuel from animal and molecule, of course. Does anybody have questions until now? Okay. I will actually go on with the lecture. I don't think it's too heavy for us to take a rest. Okay? So, next part is going to be talking more about the more academic, or what will we study and how will we study it. We finished talking about how the science is organized and how it originated. And I hope it was kind of interesting for you. But next, let's talk about something very important. This molecule, you all know it. This molecule is super important in biochemistry, in cell biology, molecular medicine, and medicine as a whole. ATP, it is the energy currency of the cell. The word currency is a perfect way to describe ATP, because ATP isn't the best way to store energy. It is the best way, it's the most convenient way to send energy from a place to another place, and then make it be used as quickly as possible. That's what ATP really is, okay? And that's why, when I hear a lot of people say, oh, we turn glucose into ATP. That makes no sense. We don't turn glucose into ATP. We extract energy from glucose, from other biological molecules, and put them in the bonds between phosphates in the ATP. These three phosphates are like a triplet. They hate each other, and they want to be off of each other. And that's why it's very easy to break them and get the energy between them. Why do they hate each other? Well, they're all negative. That's it. You can understand that any way you want. Anyway, ATP, adenosine, a ribose sugar, and three phosphates. This molecule, you'll keep hearing over and over and over. I'm pretty sure all of us know it. I'm pretty sure none of us really need a course about it. But, food. Biochemistry actually studies food. What do we do with food inside our body? That is biochemistry. That is it. I don't think that throughout this entire year that we studied biochemistry, there was a single lecture that didn't deal with food. Okay? Now, food, once you eat it, you process it heavily. Inside your stomach, of course, you process it, but also inside of your cells. You process each one of them. Each piece, you break it down more and more and more until you can use it more efficiently. That's food. These processes that you do on these cells, on these molecules, are named metabolism. This is what we do to our food. We metabolize it, okay? Now, metabolism is reactions, the reactions we do on food. And from that definition, or that simple, very simple definition, you can get two parts of metabolism that you will study in biochemistry as a whole. You will study the reaction, and you will study the reactant. That is all you will study, basically. And I will expand further on this in a bit. However, again, as I said, biochemistry studies food, biochemistry studies metabolism. And with metabolism, everything related to metabolism. So the reaction and the reactants. Now, let's talk about food. Food in a biochemical context is subdivided into two parts, two categories. We have oxidizable food. This is the types of food that we can deal with. We can change, we can break, we can do whatever we want with. Does anybody have an idea what would be put under oxidizable food? Go ahead. Carbohydrates, that is correct. Anyone else? Go ahead. Lipids, that is also correct. Yes? Proteins, correct. Lastly? Yes? Perfect. That's it. Oxidizable foods. Now, we usually don't consider nucleic acid as food, but, well, we're eating cells, and meat cells, plant cells, they do have nucleic acids. So yes, especially meat, or red meat, is very high on a type of nucleic acid called purines. Okay, just okay. So that's our oxidizable food, the food we can deal with, change as much as we want. Next, we have the non-oxidizable food, and then vitamins, minerals. Lastly, perfect. That is our food. This is everything we will touch upon in biochemistry. Now let's go one by one. Carbohydrates. It is a polymer. The monomer is monosaccharides, one sugars, unisugars like glucose, for example. Okay. Now let me ask, functions. Does anybody know functions of carbohydrates in our body? Yes? Source of energy. Perfect use of words. A lot of people will say it stores energy, and that is incorrect. It is a store, it is a source of energy. Storing carbohydrates are very, very inconvenient and very bad at storing energy. You can store energy in them that will only barely last you like 8 hours as glycogen. Okay? So it is a source of energy. Any other idea? Another function? I don't see it. Go. And what? Perfect. Immune system. That's actually one of the things I've written here. And there's a third one. It is very similar to the immune system actually, and it is cellular interactions as a whole. So it can be a messenger in cellular interaction. It can be, yesterday we took in the cell lecture with Mohammed Jamour, we took that the phospholipid bilayer it actually has some carbohydrates that are sticking out of it. Those can be used as an identifying point for these cells, and thus communication between the cells can happen just because they have a carbohydrate outside of them. Okay? Lastly, as she said, involved immune functions. Okay? Because a lot of antigens are carbohydrates. For example, the ABO system in our blood, those are carbohydrates. Okay? Next, proteins. Mainly in meat and beans, red meat and beans, or all types of meat. The monomer would be amino acids. Now, when you ask someone what are the functions of protein, the real question should be, what does the protein not do? Proteins are versatile, super versatile, variable, unique. There are so many functions that are done by proteins. You might as well be just a walkie-talkie pack of proteins. Okay, it is so important in your body, and it has so many functions that I can put them in just one slide, 2 slides, or a lecture. Okay, but let's talk about the main ones. Any idea? Go ahead. Translates DNA into RNA.
No, translates DNA and translates DNA has matter and it translates it.
I'm not sure how to understand that. Maybe you need to think about it again. Go ahead. Enzymes. Okay. There is a natural catalyst. Someone here. Go ahead. You. On the right. Hi. Yes. It is a structural component, as we studied yesterday with Mohammed Jamour, actin and myosin in the cell. Go ahead. That is exactly what structural component would mean, that they are a structural component of the muscles. Perfect. Yeah. They help with transport because, as Mohammed Jamour also said yesterday, we have transport proteins inside of our membrane, transport proteins. Okay. Messengers. Can you give me an example? RNA is kind of, yeah, but it's not a protein. RNA isn't it, right? RNA is a nucleic acid. Yeah. Okay. But no, it's not a messenger here. It is really a structural component of ribosomes. And it's a good example. Go ahead. How so? Hemoglobin. Good. It is a part of hemoglobin. Hemoglobin isn't exactly a part of any, messenger, but like the main messengers, if someone tells you, oh, messengers, protein messengers, talk about hormones. Hormones, all of them. Hormones are proteins or derivatives derivatives of proteins and amino acids. So adrenaline, insulin, hormones, proteins. Okay, what else? More functions. Go ahead.
Glycoproteins, cell receptors.
What?
Glycoproteins, cell receptors.
Okay, that's is that just a structural component? I think so. Anything else? Okay, that is a part of what a messenger does. And that is a function of protein, of course. As I said, function of protein, endless. Go ahead. That is a structural component, again. But yeah, space and components. Go.
Neurotransmitter.
Neurotransmitters, messengers, as we said. Go. They what? Good. That's true. They bind with lipids to make them mobile. They are used for transport. Yeah. I think I don't see any hands. That's fine. I said they have a lot of functions, but I wrote like around 5 functions that I found main here. They are involved in immune reactions. Antibodies. Antibodies are almost 100% proteins. My seniors will know that better than me, honestly. But yes, antibodies are mostly proteins. Messengers and cellular interactions, as we said, hormones. Hormones are proteins or amino acid derivatives. Okay? They are a structural component, as we said, actinomycin in the cell, and the most abundant protein in the body. Does anybody have an idea what it is? Just shout it out. I did not hear it once, but maybe someone said it. It's collagen. Collagen is so abundant in the body. Basically, 90% of your proteins in the body are actually collagen. They are a structural component of your skin. They are the reason why when you do this, your skin doesn't tear out. They are a structural component of the bones. They are the main reason why your bones don't really break apart. Okay? And collagen has many types, of course, but like it's 90% of the protein in your body. What else do you want me to say to tell you how important it is? Next, we said help in transport. He mentioned, some people mentioned hemoglobin, some people mentioned the lipids, but it also helps in storage. Like for example, let's say iron. Iron is not very good when it's like really free inside the cell, okay? So we need a protein that will hold this iron. It's called ferritin. It just holds it. It keeps it in place. That's it. That's all it does. For example, okay? It stores iron so that it doesn't react with everything else around in the cell. Lastly, and definitely most important for the context of biochemistry, they are natural catalysts, enzymes. And I'll not really talk much about it right now. I have a full slide on it later and 2 lectures for you guys in 2 weeks. Just this. So, next, lipids. Just a sec. Monomers are fatty acids. We all know that. They found in oils. They are either saturated or unsaturated. Go. Store energy. Perfect use of words. Oh shit. Okay. You. Yeah, she said that. It's fine. No, go.
Hormones.
Hormones. Steroids. The word is steroids. But yeah. Go ahead. Storage of some vitamins. That's perfect. I actually didn't write that. Now that I remember it, yes, there are vitamins that are stored in fats, like A, K, E. Go. Yes, you.
Phospholipid bilayer.
Perfect. Cell membrane. Any other? You.
This gives some source of external insulation.
External insulation, so kind of protection. Yeah. That is mostly what I wrote. First of all, messenger interactions, as I said, steroids. What are steroids? Like what? Testosterone, estrogen. Both of these are derivatives of fatty acids, of cholesterol, actually. Okay? Store energy. Carbohydrates are a source of energy. And lipids are a storage of energy. You store energy inside them as adipose tissue, of course. They are an important component of cell membranes. As we studied yesterday, and many of you mentioned, it is we have the bilayer. Phospholipid bilayer. And that word lipid means this, exactly. Lastly, it protects a protection of internal organs. For example, say your kidney. Your kidney is surrounded by 2 layers of fat, actually. And if these layers disappear or are used up, while, for example, you lose a lot of weight in a short period of time, a lot of that fat might go away. And you don't want that fat to go away because it's very important for the kidney. It might damage the kidney. All of your internal organs are mostly covered in fat. Your heart is covered in fat. Your stomach, your intestines, all of them are covered in fat because they are perfect insulators, as we said, but also they are shock absorbers. Okay. That is lipids. Next, nucleic acids. The monomer of nucleic acids are nucleotides, which are a nitrogen base, a ribose sugar, and a phosphate. Okay. The most 2 important or famous nucleic acids are, what is this? And what is this? Perfect. Now, the functions of nucleic acids come from the functions of these 2. So, the function of DNA. Go.
No.
Yeah, it transmits but also stores. Stores and transmits genetic code through generations. The function of RNA. Go. Yes, that is actually a very interesting point. Enzymes mostly are proteins, but there is one enzyme that I know of that is called a riboenzyme because it's actually RNA that works as an enzyme. I didn't write that here because it's super, in details. Go ahead. Yes. Okay, it can be used in fingerprinting, but that's not. Yeah, DNA fingerprinting, you mean? Yeah. Yeah, but that is more like in-depth function of it. Not in the human body. Exactly. I want something in the human body. Go. Makes proteins that's used as blueprints. The RNA are used as blueprints for protein synthesis. Now, one last function. Go ahead. DNA expression? Gene expression. Stores and transmits. And the protein synthesis, both of you together work on gene expression. So that's correct. Go. Perfect. Nucleotides alone are used alone as energy currencies. ATP is an energy currency, but not only ATP. CTP, GTP, UTP, all of these can be used as energy currencies. Yes, ATP is the most used, but during your molecular medicine course, you will learn about some stuff that can only happen with UTP around, with CTP around, with GTP around. TTP, I've never seen it in molecular medicine because for reasons you will find out later, but maybe it does happen somewhere. I don't know. I'm not sure. Can't say yes, can't say no. Enzymes. Now, enzymes. Let me go back a bit. So, metabolism. Back to metabolism. We studied the 4 biological compounds, molecules, yes? Those signify or dictate the reactants of metabolism. When we talk about the reactions in metabolism, what we talk about is enzymes. That's it. Enzymes are what really pushes, what really causes the reactions of metabolism to occur. Okay? So metabolism is 2 parts. The reactant, which are the biological molecules we just mentioned, and the reaction, which is mainly caused by its enzyme. So, enzymes. What are enzymes? Proteins. That's it. They are proteins that are used as catalysts. As you mentioned before, yes, some RNA can be used as enzyme, but that is very obscure. Proteins are the main catalysts. Of course, I think all of us took in chemistry catalysts, how they work, right? You will listen more about it in 2 weeks with my friend Ijaz when he talks about enzymes. You'll have 2 lectures about it, and in these 2 lectures, he will, in the second lecture exactly, we will talk much more about this word. See, enzymes aren't only something that makes everything go faster, they make it controllable. You can control it as much as you want so that it doesn't go too fast nor too slow. Because think of it, if enzymes would work continuously, making every reaction faster in your body, the moment you eat something, the moment it reaches to the cell, it will be used immediately. You won't be able to store anything. You won't be able to use anything efficiently, actually. You will probably crave food all the time because, well, everything you eat is being used immediately. That's why the controllability of the enzymes are very, very, very important. And when we are talking about metabolism, for example, I will talk to you about glycolysis, which is a metabolic pathway. When we talk about metabolism, what we focus on the most are enzymes and how they are controlled in that specific metabolic pathway. Okay? So, again, I think all of us heard in high school that, oh, if we didn't have enzymes, the reactions in our body would take hundreds of years. And that is correct. They take so long to happen without enzymes. That's why we have them, and that's why they are super important. Go ahead.
If someone can it be a dysfunction that someone has enzymes that are too fast?
Yes, definitely. You will study it in molecular medicine. You will study a lot of enzyme problems that either they're working too much or too little, or they aren't working at all. And those are like 90% of the diseases you will hear in molecular medicine are just diseases in enzymes. That's it. You have a question?
Enzymes works
Yeah, mostly. If it works quickly and uncontrollably, yes, you will probably die. Most likely. Okay. What about the non-oxidizable foods? Well, non-oxidizable food, as we said, minerals and vitamins mostly, also water, are used. The reason I put enzyme before this is that they are used as what we call a cofactor or a coenzyme. What are these words? You will learn more about with each aspect. But basically, they assist enzymes in their functions. So the controllability of enzymes actually kind of come from them as well. Like you can control enzymes more when you have these on because most of them are activators of enzymes, but some of them can be considered inhibitors. Okay? When we talk about minerals, of course, we talk about the good minerals mostly, but you have to remember that lead or sauce is also a mineral, but it is bad for you. So not all minerals are good, and no mineral is good in abundance. Nothing is good in abundance. Okay? Don't eat too much. Don't take in too much. Vitamins, don't take in too much. Especially things like, for example, vitamin A, pregnant women, horrible, for example. Okay? You will study way more on this. You will get it drilled in your head. But again, nothing in abundance is good. Okay, we have 2 more slides to go. This will be our blueprint of studying biochemistry. Again, I'm talking a lot about biochemistry, and I kind of ignored cell biology for a long time. And as I said, it's because cell biology is studied mostly in physiology rather than here. But I will have a slide at the end about cell biology of it. But let's talk about biochemistry. So as we said, in biochemistry, what do we study? Metabolism. Okay? So to study metabolism, again, we have 2 parts, the reactants and the reaction. The reactants are very large. A lot of lectures are needed to take them on. And that's why we start with the reaction part. We study it a bit so that we can enter every lecture with a mind that is ready to take it in. And that's why to study reaction, what do we need to study? Enzymes. So we will start off by studying enzymes. But enzymes are proteins, so we kind of need to study proteins. And that's exactly what we do. We start off by the basics, amino acids and proteins. I actually looked at multiple universities just to check if all of them go through the same process I will mention to you right now. And they all do. They start off with amino acids and proteins. After studying amino acids and proteins, they go directly into enzymes. Enzymes and their kinetics. They don't delve too deep into amino acids and proteins, just enough for you to understand what an enzyme is. They go in with enzymes, with their kinetics. And this, you will take and lecture about the enzymes and lecture about the kinetics in this program. After that, you're free to go. You can study anything in biochemistry and you're ready to do it. So, you start off with biological molecules. Biological molecules, as we said, we will study carbohydrates, we will study lipids, we will study amino acids and proteins again, and you will study nucleic acids. Each one of them, you will study 2 main things about them: their structure and basics, and their metabolism, degradation and synthesis. Lipids, also structure and basics, degradation and synthesis. Amino acids. Well, we already took the structure and basics, so let's go directly into metabolism. Okay? And let's say nucleic acids, the same thing. We will study the same formula over and over and over. This sounds simple. This is simple. But each one of these lines is basically 7 lectures. Okay? You will take a lot about each one of them. And actually, our university, because it mixes cell biology with biochemistry in one course, kind of cuts off a bit. Like I've checked the biochemistry program in Alhomam University, and they have a full year just on biochemistry. What we usually study in 3 lectures, they will take it in 5 lectures. Okay? So there is much more to what you will study right now. There is always more to take. Okay, that is biochemistry. This is what you will study. This is it. Every lecture that you will take, you can go back to this slide right now and put it onto one of these bullet points. I can put them right now. I can open every single lecture I took last year and put it under these bullet points. Okay? What about cell biology? I kind of really skimmed over it. So cell biology. As I said, structure and functions. Structure and functions inside the cell. It kind of has a gray area between it and biochemistry, honestly, yeah? Because like a lot of the metabolic pathways that go in biochemistry are, well, functions of a cell, so it's kind of cell biology. And that's true. Biochemistry sometimes is a part of cell biology. So is physiology a part of cell biology? So because, as I said, we take a lot of cell biology in physiology, we will take parts of cell biology only here, and that's because we can't really put them in any other places. Okay? And for example, remember my senior, Ali Mushtaq, right now, he was talking to you about cell cycle, yes? Cell cycle, he will study in cell biology. He will study in cell biology this, the processes of life that occur in the cell, okay? The process of life in here means how does the cell stay alive, how does the cell replicate, how does the cell die? Because the cell death is extremely important, and it is usually programmed, as in apoptosis. Okay? So we will study a lot in cell biology, but there is one point, one key lecture in cell biology that we must always remember, and it will come in probably multiple parts, and it will be important for you if you're interested in the molecular level of medicine. I am in love with the molecular level of medicine, as might be oozing from me right now as I'm giving this lecture. I really love the molecular level of medicine. I kind of don't care at all about anatomy. I came into this university, into the College of Medicine, as a person who's looking for what is it inside medicine that isn't medicine, exactly. I never wanted to be a clinician. I never want to be a person who stays in the hospital, saves lives. I know saving life is an amazing job, but it never interests me, okay? I want to study, I want to learn how God created this perfect machine. And that's what I love. And that's why molecular medicine is always something that I will adore. And I will miss throughout the coming years, actually. I digress. Process of life. The lecture I was teasing about right now, cell signaling. Why is cell signaling so important? Well, a lot of time in physiology, you will listen to, oh, this hormone is secreted, it goes to this cell, there's a function. And there's like a gap in the middle. The hormone comes in and the function comes out. What happens in the middle? What happens in the cell? And that is cell signaling in a nutshell. Cell signaling is all about, okay, here comes the enzyme, or here comes the hormone, it touched the cell. What is the cell's reaction? What you study in cell biology is this, the endpoints of all signaling pathways in the human body. You will study everything that the cell does in reaction to any other, well, signaling pathway you will study in physiology, and embryology, in all of it. Like cell biology just covers it all by itself, okay? A lot of the functions in your body happens from the same push, okay? For example, like you have, say, I think we all know the sympathetic system and parasympathetic system, yeah? Sympathetic system works on adrenaline, yes? Adrenaline itself can come into a cell, a muscle cell, and cause contraction. It can go to another muscle cell in another area of the body, and it will cause relaxation. How does that happen? Well, cell signaling explains it all. Okay? You won't go into too much detail about this part in our course, molecular medicine. But if you are interested in it, you can always learn more about it by yourself and enjoy it, honestly. Molecular medicine is hated in this university a lot. I hated it, honestly. In the first semester, half of it, I hated nothing more than molecular medicine. Why? Because I didn't understand it. And this is typical for humans. You don't understand something, you hate it, you fear it. Yes? But it dawned on me, I think, it was while I was studying for the final of the first molecular medicine. While I was studying for the final, I fell in love with the subject. It made so much sense. It was so interconnected with everything we were learning that for some reason I just found myself like, how was I so stupid to hate it? Like, what did I hate it? The main points that you might hate in molecular medicine is the switch between biochemistry and cell biology. It disrupts your focus. A lot of time you'll be going really well in biochemistry. Okay, you're understanding. Suddenly you get a lecture in cell biology that is kind of related to what you're studying, but at the same time, the way you need to understand it, the way you need to approach it is vastly different. And that's why a lot of time you'll find yourself disconnected in cell biology lectures rather than biochemistry lectures. Because as I said, biochemistry is simple. It's just this. Nobody can mess up in this. But at the same time, people really mess up in biochemistry. Our batch had an average of F in the midterm. Okay? We messed up a lot. But still, I had fun. Okay? Biochemistry is fun if you take it for real. You take it as it is. Study it daily. It will be throughout your whole year. You have time to study it. Each week, you have 2 lectures of biochemistry. Say it's 30 slides. What's that? That's nothing. You take 40 slides anatomy, 50 slides anatomy. Don't just push biochemistry to the end. It is a main part of your stay in the first year of College of Medicine. If you study it weekly, you catch up on it as much as you can. I promise you, assembly questions will sound like cakewalk, as it did to me. Okay? Yeah. That's it. If anybody has questions, even though I don't think questions will come from an introduction, but if anybody has questions, I'm very cool.